The thing that my lab is studying is perhaps yet a more nuanced way of thinking about focused ultrasound, which is almost like a way of sounding the local alarm bell within the tumor. A lot of tumors are very good at evading the immune system, and essentially what we can do with sound waves is manipulate them in such a way that we're kind of rendering them like a beacon to the immune system and letting the immune system know that there's something there that's not supposed to be there.
Welcome to Curing with Sound, a podcast presented by the Focus Ultrasound Foundation. I'm your host, Allison Preston Smith. In this episode of Curing with Sound, we're joined by Dr. Natasha Sheybani, Assistant Professor of Biomedical Engineering at the University of Virginia and the Research Director of UVA's Focus Ultrasound Cancer Immunotherapy Center. Dr. Sheybani is at the forefront of a new era in cancer research, one that combines the precision of engineering with the power of the immune system. In March 2025, she became the first researcher at UVA to earn the Era of Hope Scholar Award from the Department of Defense's Breast Cancer Research Program, a $5.5 million grant towards her research harnessing focused ultrasound in the treatment of patients with metastatic breast cancer. Before launching her lab at UVA, Dr. Sheybani earned the NIH Director's Early Independence Award, recognizing exceptional young scientists ready to lead research programs right out of graduate school, bypassing the traditional postdoctoral path. Now, she's combining that drive and creativity to push the boundaries of how sound waves can be used to help the immune system fight cancer. Welcome to the podcast, Natasha.
Thank you for having me.
What initially sparked your interest in focused ultrasound and immuno-oncology?
So my origins actually began in the field of drug delivery. I was around the age of 14 that I managed to find my way into my first laboratory setting. And I think that really sparked what ended up being a long-term interest in drug delivery. Right at the juncture of entering my PhD, when I was deciding what to do in grad school, I really honed in on this space that at the time I was reading about in broad strokes, image-guided drug delivery. As it turns out, I was stepping into the field of focused ultrasound without even really knowing it. And I remember distinctly when I interviewed with my PhD advisor, Dr. Richard Price, I basically got hooked right from the beginning of our conversations because I felt like focused ultrasound is this rare thing in medicine, actually. It's a way to precisely interact with tissue from outside the body without incisions, with a form of energy that's familiar to all of us, sound waves. So in that sense, as an engineer, it felt like I was stepping into something quite magical. Like we were turning physics, something I was very familiar with, into something that's biologically, even clinically meaningful. And then immuno-oncology, I can get into separately. I mean, that really pulled me in for a different reason. And it was actually a practical one at the time. I stepped into the field right when the Focused Ultrasound Foundation actually launched their very first immuno-oncology consortium. I had the good fortune of being part of that when several teams were brought together to essentially interrogate the effects of different focused ultrasound regimens in glioblastoma in a preclinical model. And that actually ended up informing what ended up being, my PhD focus, not just at the in the setting of glioblastoma, but also in breast cancer.
You were recognized by STAT as a wonderkind in 2020 and received several awards early on in your career. How did that recognition shape the work that you're doing today?
So the STAT Wonderkinds opportunity was totally amazing. And I first have to start by crediting, the individuals who put that on my radar. In fact, there were folks at the Focused Ultrasound Foundation who first put that mechanism on my radar. And the reason I say this is in part for anyone who's listening. You don't know what you don't know. And I actually didn't even know that mechanism existed, but I did have the good fortune of receiving the Wonderkinds recognition at a time when I was making the transition from my PhD to my postdoc. I was doing a lot of soul searching at the time. I was obviously very embedded by that point at this intersection of FUS and immuno-oncology. But in really thinking at the time about what I wanted to do with my future, that recognition was really validating, honestly. It was also just very surreal. And I think it kind of raised the stakes, actually, like in a very healthy way. It reminded me of kind of what it means to have a platform and what one can do with that. And so at that moment is when I really decided that, I want to tackle problems that matter. I want to take risks and hopefully eventually, inform the collective mentality, right? Like build the teams and the systems that would outlast me in this endeavor.
In March 2025, you were awarded $5.5 million in funding from the U.S. Department of Defense Breast Cancer Research Program through their Era of Hope Scholar Award. Congratulations on that incredible achievement and being the first recipient of the award at UVA. Can you tell us about the research project and what it entails?
Absolutely. So let me start by just expressing how incredibly honored I was and we are as a lab to have been awarded the Era of Hope Scholar Award. We were very fortunate, especially in this time of, the funding climate being what it is, that the only award DoD gave out last year went to our lab. And with that, I think comes a tremendous responsibility to do right by the community that is funding us. The award through the Breast Cancer Research Program is essentially centered on applications of focused ultrasound for breast cancer in the primary setting, as well as breast cancer in the brain metastasis setting. And across the board, the high-level question of the project is, can focused ultrasound actually make metastatic breast cancer more treatable by immunotherapy? If you look at the breast cancer landscape, the waves, no pun intended, that have been made by focused ultrasound are largely limited to thermal ablation. Essentially, what we're hoping to do is understand if we can render breast cancers more responsive, treat them more precisely, and potentially reduce toxicities, not just associated with novel immunotherapies, but even conventional modes of breast cancer care. Overall, we're kind of combining three general ideas in this grant. One of them is the use of focused ultrasound as a local kind of immune primer, which we already talked about. Using focused ultrasound to improve not just penetrance, but distribution of complex macromolecular biologics into these breast cancer settings. And finally, and perhaps most importantly, we're really trying to bring a vision to bear on the non-invasive monitoring approaches for personalized treatment. So again, we've kind of planted our flag in the spaces of PET imaging, where we've actually developed first-in-class novel instrumentation for integrating PET with focused ultrasound, even under MR guidance. And secondarily, we're also doing exercises in biomarker discovery at the level of liquid biopsy. So again, imagine that I can glean something about the way your cancer is evolving or responding to therapy from a blood draw. The vision is really now to narrow in on breast cancer as the space to realize that. One other thing worth noting is we have, again, a very ripe translational pipeline here at UVA that has already given rise to multiple clinical trials in breast cancer. So part of our goal with this grant is also to really interface even more with the clinical enterprise. We intend to hopefully realize what might be future clinical trials as well by virtue of the work that we're doing at the bench side. And of course, where we're already treating breast cancer patients, we're really going to make a bona fide effort to understand everything we can from the work being done there. And finally, a pillar that really informs all of this that I mentioned is the breast cancer patient advocates who we were fortunate to actually bring in as the grant was being written. So they really informed the vision for this grant. Essentially, I had a group of women who were my sounding boards to say that what we're doing is, palatable, or, it's something that I would never choose to do. versus, this was my experience and I really wish I had had a better option in this specific sense. So like I did a lot of due diligence as I was writing the grant with this team of patient advocates. And now our goal over the next few years is to actually build out a sort of coalition of patient advocates who will inform this science, but also other sectors of science that we hope are inspired by this work.
That's such an incredible way to draft the grant, to get input from women who have been through this experience. Was there anything surprising you learned from that?
Yeah, I just, it should come as no surprise, but I feel like what I learned was that format can exist, honestly. Like as researchers, we're so often in our own silos, and the barriers between us and the community are sometimes a bit more significant than we even might realize. That barrier was actually incredibly easy to break down in this process. And it just goes to show how simple it can be to sort of, reach out and get quite a lot back. These women have been so generous with their time, with their insights, with their vulnerability to share like the totality of their experiences undergoing cancer care. But like I learned that model can exist, and there's no reason that we only have to implement it here. And I think the other message I want to get out to other scientists is that this is a model that we can all stand to follow more. So that the work that we're doing is that much more accessible and that much more translationally relevant when all is said and done. So that might have been the biggest lesson. And again, the long-term hope is that in lieu of just propagating that message, we can actually establish an infrastructure within UVA that people can tap into to take advantage of, opportunities to engage with the community.
Could you explain in lay terms how focused ultrasound is being used as an immunomodulator in solid tumors like breast cancer?
So the thing that I often say when I'm talking about focused ultrasound is that we're essentially using the physics of sound waves to tune biology. And often, especially in breast cancer, when people think of focused ultrasound, they think of the mode known as thermal ablation. This idea that we can essentially use heat to destroy tumors. There are a lot of other things we can do, though. And that's sort of the preface here as I talk about FUS as an immunomodulator, because in one respect, we can use focused ultrasound to stir up the tumor microenvironment, not just with heat, but also through mechanical regimens. Histotripsy is an emerging and very relevant example these days. Moreover, we can use focused ultrasound to kind of loosen physical barriers that might prevent our therapeutics from entering these tumors. And finally, the thing that my lab is studying is perhaps yet a more nuanced way of thinking about focused ultrasound, which is almost like a way of sounding the local alarm bell within the tumor. A lot of tumors are very good at evading the immune system. And essentially what we can do with sound waves is manipulate them in such a way that we're kind of rendering them like a beacon to the immune system and letting the immune system know that there's something there that's not supposed to be there. And this is one of the primary issues with immunotherapies, is that while they can enact really powerful effects against the cancer is very good at essentially masking itself. So in that sense, that final pillar I mentioned is sort of the classic way that we're thinking about immunomodulation.
How do you decide when you've treated enough with focused ultrasound, and how do you monitor effects on immunotherapy, tumor microenvironment, and patient response?
Yeah, I mean, I think that the question you're asking about when have we treated enough? So at the end of the day, I think that this is a question that we have the ability to tackle in unique ways with focused ultrasound. But again, we're not really the only ones thinking about this. And I preface with that because the tools that my lab is using to think about that question borrow deeply from other fields. So namely, when we're thinking about have we treated enough, my first criteria for making that assessment is to stay true to the benefit of our approach. A major benefit associated with focused ultrasound is the fact that it is non-invasive, the fact that it can be done repeatably over time. And so with all of that, at least in my lab, we decided to kind of hold our surveillance methods to this very stringent criteria that they had to be the same. They had to be sort of non-invasive, longitudinally accessible approaches. Now, if you look across the spectrum of things that we have access to, the things that kind of rise to the top would be approaches like imaging, right, where we can sort of monitor the tumor non-invasively, as well as liquid biopsies, for example. Any place where we can draw from a bodily fluid, say the blood, and understand something about the analytes that relate back to the tumor, either in its evolution or in its response to therapy. So as we are deciding on whether we've treated enough, we're really just thinking about ways that we can integrate more effective, higher fidelity surveillance approaches with our focused ultrasound techniques, with the goal being that if we are able to collect those measurements, there might be sort of unique biomarkers and interesting signatures that we can begin to associate with the responses to focused ultrasound. Those biomarkers could one day potentially inform the way that we deploy therapies, the way that we choose specific combinations. They potentially could inform the way that we adapt therapy over the course of intervention. And coming back to that question of what is enough, I think that we're really going to need to make those assessments in order to understand where we've been effective, what did it take to be effective, and how well can we reproduce that.
How might this change the patient experience in terms of side effects, recovery time, or outcomes?
I hope that one day we have the capability to be totally transformative. So at the risk of being a little too provocative here, I would say that if we do this right, there could be profound impact for patients. And in practical strokes here, I would say a few things. One is, again, we're talking about the potential for less invasive treatment. Something that could even be an outpatient intervention. Again, we talked about the principle of like no incisions, no catheters, none of the sort of other cumbersome aspects that come with your typical cancer intervention. We're talking about the potential for faster recovery times, even the potential for, without it being too arduous, repeat sessions of therapy if needed. And most importantly, even our best cancer therapies still come with pretty harsh side effects. So something that I'm looking at closely right now, even in the context of immunotherapy, is where we still have toxicities associated with our novel or conventional cancer therapies. So the potential to really de-intensify some therapies, especially the ones that are administered systemically, and thereby offer a safer but still effective option for cancer patients, I think is huge. And in this sense, I also want to be careful not to overpromise, right? These are all things that we are interrogating in the lab presently, with our North Star really being, can we make immunotherapies much more effective than they are presently, but also think in tandem with that about the situations where therapies, immunotherapy or otherwise, are effective, but could be made much safer, much more accessible and comfortable for patients.
When it comes to your lab, you have included students at every level, middle and high schoolers, undergraduate, graduate, postdoctoral, even medical residents. What drives your commitment to mentorship, and how do you approach guiding the next generation of researchers?
Honestly, I have to say, mentorship is my favorite part of the job. I'm supposed to say, like the science is so exciting and I love it. But I think getting to spread that excitement to others and seeing their passions get sparked as trainees is everything these days. Mentorship is a multiplier, right? So in a sense, I view a large part of my charge these days as a PI through that lens, inspiring others to also think about these problems, develop the real like passion for ushering critical engineering problems forward and thinking about solutions. And, you know, I think getting to also experiment with how to do that in the lab has been exciting because you mentioned, you know, the kind of composition of the lab. I also think that, you know, being at the University of Virginia in particular, it's a very cool experience that, of course, I got to have being mentored and, you know, training within this environment because we have, particularly for focused ultrasound, a really ripe, again, translational ecosystem. We are also now building out sort of a linkage to the community within my lab where we're engaging with breast cancer patient advocates as part of the work we're doing in the breast cancer space. And so the students really get to sit at the center of all of that. And it's not difficult to inform the why when you get to interact with everybody from the basic scientists to the clinicians to the patients and even the Focused Ultrasound Foundation, which is right down the street. So in this sense, there's been such really a richness to the mentorship experience. And again, one of my favorite parts of the job.
What do you hope your work will make possible for the next generation of patients and scientists?
So I'll answer those again separately. I'd say for patients, what I would fundamentally hope that the work means is just more options, right? Especially in situations where patients are unable to benefit from standard therapies or are faced with, again, the limitations of, you know, invasive risk, toxicity, et cetera. My hope is that we can carve out a path to treatments that are effective, but again, far less punishing when it comes to the side effects. I'm emphasizing this because I think it's been so illuminating for me and for us as a lab to actually engage, for example, with women who have themselves undergone breast cancer treatment and very fortunately have been survivors of the disease. But now you look at where they are at, even 10 years out from that experience. And the thing we don't talk about enough is survivorship. We don't talk about all of the detrimental downstream impacts of even surviving cancer. So I think it's not just the treatment, but it's also the long-term implications of the treatment that we want to be critically aware of, and where there are truly low-hanging fruit opportunities, I would say, for focused ultrasound technology. For scientists, I really think of focused ultrasound as a platform technology. So there are many possibilities I could point to, and I would say, like, my hope for the future is that we are really bringing together all corners of the scientific domain to tackle problems in an interdisciplinary way, to ask new questions and conceive of new therapies that haven't even been thought of before. I think what I have witnessed over the last 10 plus years of being in this field is this, you know, huge evolution in the backgrounds and the sort of schools of thought that have been brought to the technology. And that's where I think we're really now seeing an inflection point and accelerated growth.
Well, you have a lot of exciting research projects going on. How can listeners keep up with your work?
All kinds of ways. So I would say the best way to keep up with our work is through our lab website, sheybanilab.com, through my updates on LinkedIn, where you can follow me, and on X, though I'm admittedly less active there. And of course, my email is sheybani@virginia.edu. We do also have a Focused Ultrasound Center of Excellence and our Focused Ultrasound Immuno-Oncology Center at UVA, which folks can also stay updated through. And of course, through our publications and the opportunities where we're stepping into the Focused Ultrasound community. So we welcome folks to reach out anytime.
Well, thank you so much. This has been so great talking to you, hearing about your important research going on, and thank you for all your important work.
Thank you so much for having me. It was such a pleasure.
This episode of Curing with Sound has been presented by the Focus Ultrasound Foundation. If you'd like to learn more about Focus Ultrasound or the Foundation, visit our website at fusfoundation.org. You can also sign up for our newsletter and follow us on social media. Be sure to subscribe to the Curing with Sound podcast so you don't miss the latest information about Focus Ultrasound. You can also rate and review the show.